Hi dear students, a warm good morning to all of you. Welcome to the second session of Immunology. Previously we have been talking about the innate immune system or the self-immune system which has been developed when we are being born itself. By birth itself we get an immunity, and that is referred to as an innate immune system, and that has been contributed by various physical, chemical, as well as physiological, as well as various cellular factors. Previously we had mentioned about the discussed about the physical factors as well as the physico-chemical factors, which will physiological factors as well as the chemical factors, as well as the microbiological factors like resident bacteria also contribute towards the innate immune system. Now in this session, we are going to talk about the cellular factors which will contribute towards the innate immune system. If you go to look onto your immune system, okay, especially the innate immune system, you can see that different cells do contribute towards your inborn immunity. And a major amount of this immunity is being contributed by the cells which have been present within your blood. And within the blood, we do know we have RBC as well as WBC. RBC is the most predominant component, as we all know, which contributes to the red color of a particular blood. But the white blood corpuscles also play a very important role in the innate immune system. And they do occur in different forms. And among the WBC, you can see that various components like the basophils, eosinophils, neutrophils, then various other components like you have monocytes and macrophages and all that. Monocytes and macrophages, they contribute towards the innate immune system. If you divide the blood cells, you have RBC, WBC, and platelets. RBC is, RBC and platelets, you keep it aside. Next, you have the WBC. Among the WBC, the one nucleotide cell is called the monocytes. And the monocytes will further differentiate in the tissues to form the mast cells. So, monocytes and mast cells that we have, not the mast cells, the macrophages. The monocytes will differentiate into the macrophages in the tissues. So from the blood, you have the WBC cells, the basophils, eosinophils, neutrophils, as well as the monocytes and the macrophages, which contribute towards the to the innate immunity. And you can see that other cells, like you have Langerhans cells, the mast cells, they also they also contribute towards the innate immunity. And you can see that some cells called the natural killer cells, as well as the one member, that is the gamma delta T cells, they could also be considered to be as members of the innate immune system. Now this is a distinct figure which I have taken from the Nature reviews. So you might not come across the gamma delta T cells quite frequently in your textbooks, but these two, that is a NK cell, is being found to be more common in the innate immune system, where you get a rapid response. So NK cells and gamma delta T cells, they are usually considered to be as they are lymphocytes, okay, and they do have some antigen specificity also sometimes, and so they might be considered to be innate cells with some similarities to the effective lymphocytes. And you can see the other cells, okay. Now B lymphocyte and the T lymphocyte, lymphocytes, the B lymphocyte and the T lymphocytes, they are being mainly involved in the adaptive immunity or the acquired immunity. So in the innate immunity, you do have all these cells. The NK cells, the basophils, eosinophils, neutrophils, then dendritic cells, mast cells, macrophages and all that. But since some NK cells as well as the gamma delta cells do show some antigen specificity, sometimes we might feel that they are also in adaptive also in nature. So in this class, we will be talking about the different cell types and what are their roles. So that's what I have depicted over here. the NK cells can go sometimes commonly to for both adaptive as well as innate. But all these other cell lines, neutrophils, eosinophils, basophil, mast cells, monocytes, dendritic cells and all that, they all together contribute towards the innate immune system. So let's move on. First, coming to the single nucleated cells which have been found within the blood. They are called the mononuclear phagocytic cells, and they include the monocytes which have been circulating in the blood and the macrophages which have been deposited within the tissues. So macrophages, they are considered to be 5 to 10 fold larger than the monocytes, and they contain organelles, especially the lysosomes. These, as a term implies, you can see mononuclear phagocytes. They are cells which do take part in the what? Phagocytic process. They are mononuclear leukocytes with an ovoid or a kidney-shaped nucleus, and they do have granules within their cytoplasm. And they are mainly being produced in the bone marrow, and they enter into the blood, circulate for 8 hours, enlarge and migrate to the tissues, and they can either mature into the macrophages or into the dendritic cells. You can have a look here. The monocyte cells, either they mature into the macrophages or they mature into the dendritic cells. And they do carry out the process of phagocytosis. Now, what is a macrophage? This is a macrophage which is, you can see. And you can see macrophages, they are being dispersed throughout they are not found in the blood, they are mainly found in the tissues. They are larger than monocytes and they contain more organelles that are critical for phagocytosis. That is, for phagocytosis, you require large amount of lysosomes and all that. So, such organelles are being found to be more in the case of macrophages. And sometimes they take up residence in a particular tissue, becoming fixed macrophages, whereas sometimes they might be free or wandering and all. And the free macrophages, they travel by an amoeboid movement throughout the tissue. And you can see that this macrophage, at different places, they are being named as different. Sometimes in the liver, they are called the Kupffer cells. When they get deposited in the connective tissue, they are called the histiocytes. In the kidney, they are called the mesangial cells. And in the lung, they are being called the alveolar macrophages. So the macrophages are nothing but a differentiated monocyte which gets deposited in a tissue and which carries out the function of what? Phagocytosis. And you can see that the macrophages, they do have receptors for attachment of various components like mannose and fucose receptors, Toll-like receptors, which these receptors can bind to the lipopolysaccharides, the peptidoglycan, as well as the fungal cell wall components called chymosin. And hence, if a bacteria is coming into the blood, into the tissues, okay, what happens? These receptors which have been found on the macrophage, like the mannose and the fucose receptors, as well as the Toll-like receptors, will bind these these bacteria over there, and then they will be interacting, and phagocytosis will be carried out. And you can also see they also do have receptors for some antibodies and serum proteins called the complements also. And hence, what happens when complements gets attaches, when antigen antibody complex as well as a complement attaches, a process called opsonization or increased level of phagocytosis can be carried out. So the macrophages do play a role in the process of increased phagocytosis, which is termed as the opsonization. Now, the next cell that we have is the dendritic cell. As I told you, the monocytes can either mature to form a dendritic cell or a macrophagic cell. Now, the term dendritic cell, it has mainly been acquired because it is covered with a long membrane extensions that resemble the dendrites of nerve cells. And these dendritic cells are difficult to isolate because the conventional procedures for cell isolation tend to damage they are long extensions. And there are different types of dendritic cells. They are like Langerhans cells, interstitial dendritic cell, myeloid cells, and the lymphoid dendritic cells. Even if you don't go into the details of it, you can just know that these are the names of some of the dendritic cells which have been present. And what do? They mainly recognize the antigens by their surface molecule, phagocytose them, and then they get activated. And you can see that they also serve as potent antigen presenting cells than the macrophages and the B cells. What is antigen presenting cell? A cell which will process and present the antigen towards so that the pathogen will be killed completely. Okay, so for the lymphocytes to act and all that, okay, like the T cells and all that, for them to act, these antigens, or when a pathogen comes, it has to be uh, processed and linked with some bacteria or with some cells, and they should be presented towards the T lymphocytes. That is called antigen presenting, and so that has been effectively carried out by the dendritic cell. And dendritic cells can migrate towards the lymphoid tissue, and it act as activated cells, and they represent and they will present this antigen to the T cells over there. Thus, not only do the dendritic cell destroy the invading pathogen as a part of innate immune response, but it also triggers the specific immune responses also. So that's about the dendritic cells. And now we'll move on to the next type of cells which we have in the WBC. Like we have the neutrophils, eosinophils, as well as the basophils. The multi-lobed nucleus is mainly being found in a neutrophil cell. And if you go to consider the eosinophil, it is almost having two lobes. Okay, and basophil also, the nucleus is, so it's based upon the nucleus that you identify whether it is a neutrophil, eosinophil, or a basophil. Now, neutrophils, they are phagocytic cells with a nucleus that is having 3 to 5 lobes connected by slender threads of the chromatin. And they have inconspicuous organelles called the primary and the secondary granules. And the primary granules do contain various enzymes called the peroxidases, lysozyme, defensins, and various other hydrolytic enzymes. And the secondary granules do have various other enzymes called the collagenase, lactoferritin, catalycidin, lysozyme, and all that. And all these granules will help the foreign material to get digested after it is undergoing phagocytosis. And neutrophils, they also do use an oxygen dependent as well as an oxygen independent pathway to kill the ingested microorganisms. And just like the macrophages, the neutrophils also have receptors for antibodies and the complement proteins, and they are being found to be highly phagocytic. So the process of opsonization could also be carried out by the neutrophils. But unlike the macrophages, the neutrophils does not reside in a healthy tissue, but they circulate in the blood so that they can rapidly migrate to the site of tissue damage as well as infection, and they become a principal phagocytic as well as a microbicidal cells which have been present in the blood. So when you have macrophages which have been playing an important role in the tissues, you have the neutrophils which play an important phagocytic role within the blood of the organism. Now, basophils, they have irregularly shaped nucleus with two lobes, and the granules are, they stain bluish black with the basic dyes. They are non-phagocytic cells, but they mainly release specific compounds from their cytoplasmic granules in response to certain types of stimulation such as histamine, prostaglandin, serotonin, as well as leukotrienes. And these are compounds, they influence the tone and the diameter of the blood vessels, and hence they are considered to be as vasoactive mediators. The basophils, as well as the mast cells, they do possess high affinity for the antibody associated with the allergic response. And when these cells become coated with this type of antibody, the binding of the antigen to the antibody triggers the secretion of vasoactive mediators. So the role of it is not phagocytosis, but it mainly is a component, a cell member, which plays an important role in releasing vasoactive mediators so that the blood vessels will get what the blood vessels, the tone and the diameter of the thing will be increased. Coming to the eosinophils, they have two lobed nucleus connected by slender thread of chromatin and granules, which stain red with acidic dyes. And unlike the basophils and the the eosinophils will migrate from the bloodstream into the tissue spaces, especially the mucous membranes. And they are an important member in the defense against protozoan as well as the helminthic parasites, and they release a large amount of cationic peptides as well as the reactive oxygen intermediates into the extracellular fluid, and they play an important role in allergic reactions, and they also release such as various inflammatory mediators like histamines, leukotrienes, etc. So thus you can see that the eosinophils, they play an important role in the allergic reactions of man. Now the mast cells, these are bone marrow cells which have been differentiated in the blood and the connective tissue. Although they contain granules with histamine and other pharmacologically active substances similar to the basophils, they arise from a different lineage, and but the mast cells and the basophils are important in developing the allergic responses. So these are the cellular factors of the innate immune system, and they contribute mainly to two different processes. One is the phagocytosis, where a bacteria becomes attached. It is a process of where the bacteria or the pathogen becomes engulfed by a process, and it is a type of endocytosis. Here what happens, the bacteria becomes attached to the membrane by to the membrane evaginations called the pseudopodia of the phagocytic cell, and it gets ingested, and within the cell it combines with the lysosome to form the phagolysosome, and the lysosome degrades the enzyme and the digestive products are being left out. So the cellular factors of the innate immune system contribute towards the process of phagocytosis, as we have already explained. In addition to this, you can also see that we also have the NK cells, which we had not mentioned earlier. The NK cells they are also a part of lymphocytes. They are a type of WBC cells, and they are also a component of the innate immune system. And they play a major role in the host rejection of both tumors as well as the virus infected cells. So these are cells which have been found to be highly cytotoxic, okay, and there are certain granules within their body, within these cells, in the NK cells, and they are called as and they have specialized proteins called the perforins or the proteases, which are known as the granzymes. And the perforin, it will form pores in the cell membrane of the targeted cell, while the granzymes will mainly produce molecules which will enter and will cause apoptosis or the programmed cell death of the particular of the targeted cell. So the targeted cell here, the NK cells mainly act on the tumor cells as well as the virus infected cells. And you can see that when apoptosis, okay, and cell lysis is being found to be a little bit different. In apoptosis or the programmed cell death, what happens? The destruction of the virus inside the cell does happen. But in a normal, if you're going to consider just lysing a virus infected cell, the virions will be coming out, but only the cell gets lysed. So the effective killing of the infected cell as well as the virus by apoptosis or the programmed cell death is being carried out by the NK cell. And they mainly mediate by various cytokines, like you have interferons or macrophage derived cytokines. They all will promote the NK cell activity, and they serve to contain viral infections, while the adaptive immune response is generating antigen specific cytotoxic T cells that can clear their function also. So you can see that, as I mentioned earlier, the NK cells do play an important role in the innate immune system, but sometimes it also show some roles of the of the other one also, the acquired immune system. And in the innate immune system, we also do have another process called the inflammation. Inflammation is what is inflammatory response. The tissue damage which has been caused by a wound or by an invading pathogenic microorganism induces a complex sequence of events, and that is mainly been known as an inflammatory response. And you can see that when a tissue damage comes, the tissue damage will release various vasoactive and chemotactic factors, and that will trigger the local increase in the blood flow and the capillary permeability. And so when the tissue damage occurs also, some bacteria could also happen to enter into that. And the permeable capillary, so when the tissue damage is formed here, vasopermeability increasing substances are being released, and the capillaries become more permeable. And so what happens? From the blood, the exudates like the complement antibody and various C-reactive and all will come outside. And what happens? By the process of extravasation, the phagocytic cells will come outside through these holes, and they will migrate to the site of inflammation, and they will carry out the process of phagocytosis. So when the normal phagocytosis happens within the blood, the process of inflammation can be carried out within the tissues, and the phagocytic cells play an important role in it. And you can also classify the innate immunity as species immunity, racial immunity, as well as you also have the individual immunity. If one species is resistant to a certain infection and the other is not being susceptible to the same infection, then you call it as species immunity. Now, for example, you have the birds are resistant to anthrax, but humans are susceptible. And sometimes some races will be having a special type of immunity because they are being resistant to some infection. Now, for example, African race, they are resistant to malaria and yellow fever, whereas the Asians and Americans are susceptible to the same infection. So then we have also individual immunity. Only some people will be resistant to some diseases and all that. Okay. Now, individuals with genetic deficiency of glucose 6-phosphate dehydrogenase, they have been resistant to malaria. So I guess we have come to an end of the innate immune system and the various cellular factors. Thank you for now.